Are you a student seeking out genetic assistant positions? These vital positions aid with clinical and research patient communication, data entry, genetic testing coordination, and administrative tasks. Therefore, training is key. We recommend the Genetic Assistant Online Training Program at Johns Hopkins University School of Medicine. This online program provides knowledge and skills to learners considering genetic assistant positions or those recently hired into these roles and need job training. The program consists of 2 10-week instructor-led courses. At the successful completion of the program, learners will receive a certificate of completion from the Johns Hopkins School of Medicine and the McKusick-Nathans Institute for Genetic Medicine. Applications are being reviewed for the fall 2021 cohort, and limited spots are available. Applications for the 2022 cohorts of the program are also open. All you need is a basic understanding of science, particularly biology. Don't wait. Check out the Genetic Assistant Online Training Program at Johns Hopkins University School of Medicine now. Click the link in the show notes for this episode for more information, or go to dnapodcast.com.
We find ourselves surrounded by such complexity, such elegance.
The genes of you and me, the genes of you and me, we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hello, you're listening to DNA Today, a genetics podcast and radio show. I'm your host, Kira Dineen. I'm also a certified genetic counselor practicing in the prenatal space. On this show, we explore genetics' impact on our health through conversations with leaders in the field. These are experts such as genetic counselors, researchers, doctors, and patient advocates. My guests today are from BioVaxys, a leader in the vaccine industry. Kenneth Kovan is a founder and its president COO. And Dr. David Berd is also a founder and chief medical officer. So thank you both so much for coming on the show to dive into very timely topipics.
Thank you very much.
Thank you.
Appreciate it.
So a little background information on BioVaxys. It's you are developing a vaccine platform for viral infections and cancers, and also developing a really unique diagnostic in support to fight against for COVID-19. And what really brings all of this together with these 3 technologies is a focus on T cells, which is what we're going to be exploring in today's episode. So Dr. Berd, I'd love to start out by having you explain how the immune system typically fights cancer so that we understand this basic so that we can add more information on and dive into the details during the interview.
Sure. Well, the problem is that most of the time the immune system does not fight the cancer, which is why people have cancer, the cancer grows, and in the absence of effective treatment, it makes them sick and takes their life. When the immune system fights cancer, it's because cancer cells are slightly different from normal cells. It's surprising how similar they are, but there are slight chemical differences. And they have proteins, or what we call antigens, on their surface, which the immune system can recognize. And that usually doesn't work, but when it does work, the response is mediated by a special kind of white cell called a T lymphocyte. So T lymphocytes recognize the cancer antigens and can fight the cancer in 2 ways. One, they can directly kill cancer cells, or they can produce chemicals called cytokines, which kind of bring in a whole host of other kinds of immune cells which slow or prevent the tumor growth. So the trick in immunotherapy is to make some changes so that you shift the balance so that the immune system actually fights the cancer rather than tolerating it, which unfortunately is what happens most of the time.
And so then how does these Haptenized vaccines play a role in terms of taking advantage of how we know the immune system works, and what is that process?
So haptenization is an immunological trick. It was discovered many years ago, probably almost 100 years old. So hapten is a chemical, and there are many different haptens. There are 2 that we're working with, but there are thousands to choose from. They're small chemicals, and when they hook up with a protein, they change the protein subtly, so that the immune system can recognize the protein more strongly, or in extreme cases, can recognize a protein which otherwise is totally ignored by the immune system. So this is a perfect situation for cancer because, as I've already said, the cancer proteins or cancer antigens usually aren't recognized. When you haptenize them, you can get the immune system to recognize them. The immune system then makes a response, as I said, a T cell response against the cancer antigens, which have the hapten, and then there's a secondary response against the non-haptenized native tumor antigens, which of course are the ones that you want to target. The haptenization process can be applied to a cell, or it can be applied to a purified protein. In the case of cancer, we just use the cell and haptenize many of the cancer antigens on the cell. In the case of COVID, where we know what the proteins are, we can directly haptenize the protein and use that as the immunogen. The haptenization process itself is very simple. You just take the cells or the protein and mix it with your hapten of choice, and for variable periods of time, sometimes a half an hour, sometimes several hours, wash out the excess hapten, and you're done. So it's extremely simple process. And so we're using this approach for both immunizing against cancer, which, as I've already pointed out, is extremely difficult, and immunizing against a COVID protein, which is a lot easier. Obviously, we have vaccines, but which still is challenging to get a very strong immune response, particularly T cell response, without having side effects.
And so knowing all of this information, it really helps to be able to design these vaccines to target these cells and have the body understand how to fight this. What are the vaccines in development that are based on this process? Ken, I'd love to hear from you in terms of what you have in the pipeline right now.
Yeah, sure. Thank you. Currently, BioVaxys has two technology platforms that are based on the haptenization of cell proteins or antigens from tumors or viruses. BVX0918A is our vaccine candidate for treating ovarian cancer, and that is based on the haptenization of ovarian tumor antigens. BVX0320 is a parallel program also based on our haptenization platform that is targeting the SARS-2 virus. Other haptenized programs that we also have in development, they're not nearly as far along as ovarian cancer and our SARS-2 program, but others that we're looking at are for HPV, other coronaviruses for that matter, and other tumor types such as cervical cancer, renal cell carcinoma, and melanoma. The beauty of the platform is that we're not just limited to treating one particular tumor type or one particular virus. We've got the ability to throw the net, so to speak, very widely.
Biology? How about a knowledge of next-gen sequencing applications? Want to use those skills to influence change in the genomics market? Live on the West Coast? Like to travel? Then I found the perfect role for you. Genomic Software Field Application Scientist. This position is part of Agilent's Diagnostic and Genomics Group. Agilent inspires and supports discoveries that advance the quality of life. They provide life science, diagnostic, and applied market laboratories worldwide with instruments, services, consumables, applications, and expertise. As part of the team, you will provide scientific and technical consultation to consumers in partnership with the sales team to ensure laboratories learn how Agilent's genomics analysis and interpretation software can impact their specific use. Join their amazing team of scientists by submitting your application via the link in our show notes if you're listening to this through a podcast app. You can also access the link at dnapodcast.com. Have you been vaccinated for COVID-19? Wouldn't it be handy to have a digitized version of your vaccine record? States in the U.S. are starting to do this, with California leading the way. However, privacy concerns have been raised about the personal information being kept in a central database. Genobank.io offers digitized vaccine records without these privacy concerns. Genobank.io utilizes blockchain networks to keep track of your vaccine record without exposing your personal data. Check it out. That's Genobank.io. Again, that's Genobank.io for secure digitized vaccine record. And a lot of people have been learning more about vaccines with the COVID-19 pandemic and learning about how those vaccines work in terms of the mRNA approach of vaccines like Pfizer and Moderna. How is this different? Are there parallels to it for those that maybe understand the mRNA process? How is it similar or different?
They're quite different, but the similarities are that whether you're making an RNA vaccine or whether we are making a haptenized vaccine, everybody who's working on this so far is targeting one antigen or one particular protein on the SARS-CoV-2, which is a virus that causes COVID-19. And the name of the protein is the spike protein or S protein. So everybody probably has seen, if you look under a very powerful microscope and you look at this virus, which is of course very tiny, it has spikes coming out of it. It looks like a crown, which is why they call them coronavirus. And the spikes, in addition to being interesting, are absolutely critical in the virus doing us harm, because those spikes are the ones that attach on to the cells in the body, particularly, for example, the lung cells. And once the virus is attached through those spikes, the virus can then enter the cell and then cause all kinds of trouble, including making people very sick, causing pneumonia, and even in extreme cases causing people to die. So what we have in common with the RNA vaccine, and it's the only thing that we have in common, is that we're both using the spike protein. Rather than injecting RNA and then having the RNA get into body tissues and then have the body tissues make the spike protein, we start with the purified spike protein, which of course we have to produce in a biological laboratory, and then we haptenize it and inject it. So underlying, it's the same, but technically it's quite a bit different.
And there's been a lot of conversations regarding the safety concerns around vaccines. What are your thoughts and expectations for the safety profile of these Haptenized vaccines?
Okay, so starting out with the cancer vaccines, we have a current version of the Haptenized cancer vaccine that we're testing. But there was a previous version that I tested, and it was also tested by a previous company, which is no longer in existence. So more than 500 patients have been injected with the first generation Haptenized vaccine, mainly people with melanoma and ovarian cancer. And in 500 patients, there have been no serious side effects. In fact, the side effects are relatively trivial, just soreness and itchiness at the injection site. There have been no significant or serious adverse events. So we believe that the new Haptenized cancer vaccine that BioVaxys is developing, which is a little bit more complicated, a little bit more sophisticated, is going to have an excellent safety profile because we already have a lot of patient information. Our COVID BVX or BVX-0320, our COVID vaccine, of course, we haven't put into patients yet, so we don't know what the toxicity profile is going to be. However, we have done an animal study in mice where we got excellent immunological responses, antibody responses, T-cell responses to the spike protein by injecting our vaccine, and there was zero toxicity in the mice, the way you measure toxicity in mice. So that gives us great hope and expectation that our COVID vaccine is going to have an excellent safety profile in humans. Of course, there's proof is in the pudding, and we'll know as soon as we do the clinical trials. But we're expecting this not to be a big problem.
Yeah, it's excellent data to begin with, and just everything that you have with it. In terms of, you've talked about developing this and the process for it. Is there any of a genetic aspect to this, of looking at genetic sequences in terms of, with the cancer side, how cancer cells can have different genetics as opposed to normal human cells? Is that a part of it, or is that completely separate and you're not focused on that because you're looking at the proteins?
Well, I mean, we're not focused on it technically, but genetics is behind everything. So if there's an antigen or a protein on a cancer cell that elicits an immune response, obviously there is a genetics in that cancer cell that is making the cell produce that protein. But practically, our company doesn't need to be involved in the genetics, and we're not a genetics company. So we're starting with the proteins themselves. We're very interested in the genetics, but we don't need to get into it. We don't need to understand it. And because we're not involved with genetics, it makes our process simpler and probably cheaper.
Yeah, that's a good point, because genetics can be so complex. So if you're kind of starting at that protein level, you don't need all of that background information on what's happening with the genetics and side. Want to learn more about clinical genetics, molecular genetics, and laboratory science? Then you should check out the Genetic Assistant online training program at Johns Hopkins University School of Medicine. By taking part in the program, you will be joining both national and international learners with the same passion for genetics. Interact directly with your Johns Hopkins instructors and fellow students throughout the program. Applications are now being reviewed for the fall 2021 cohort, and a limited number of spots are still available. For more information, use the link in our show notes. If you're listening to us through a podcast app, you can also access the link at dnapodcast.com. We've all been at a restaurant where a QR code is on the table to access the menu. It's the new normal for more than just restaurants, though. QR codes are now rolling out for vaccine records. California is starting the trend, and more states are expected to follow. Instead of having your vaccine record card physically on hand, it's much easier to have a QR code saved on your phone. However, having this information in a central state database in California does raise privacy concerns. That's where Genobank.io's QR code design is innovative. The Genobank.io solution offers more privacy and does not require storing records in a centralized, hackable database. Instead, it uses blockchain technology to protect your data. An added bonus is the QR code generated is scannable by over 6 billion devices, and it's accepted by the U.S. Department of Homeland Security and border agents across the globe. To learn more about this protective QR code at Genobank.io. Again, that's Genobank.io to secure your digitized vaccine record. We've been talking about just how things are in terms of the process and development side. Ken, can you share what are the currently available Haptenized vaccines and like what that looks like? Are there any available at this point?
Kira, there's probably two parts to that question. The first being that there are currently a number of oncology programs with other companies that are based on the use of autologous cells, which is what we're using in terms of creating our vaccines, our cancer vaccines. Probably heard of it, CAR T therapy, chimeric antigen receptor. That's derived from autologous T cells, and there are some currently approved products using autologous cells. So that is becoming much more well known in the field as of late. However, the concept and the practice of haptenizing the tumor antigens or viral antigens, we are not aware of anyone that is doing this except for ourselves in this field. So it's a space that we believe that that we own. We have filed a number of patents and have a fair amount of intellectual property that locks up this space for us.
And which is so exciting because this is a new way to develop a vaccine, and we see just how important vaccines are, especially with this pandemic. We've all been impacted by vaccines. Looking at the other side of it, usually we're getting a vaccine for a prevention purpose, so to prevent us from having symptoms from COVID-19, for instance. But I was really intrigued to see that you're also developing a vaccine designed for people who've been diagnosed with ovarian cancer. Where are you at in terms of the stage of this vaccine? Ken, did you want to chime in and just provide a little bit of information on the current status of that vaccine?
BVX-0918A, which is our ovarian cancer vaccine, we're currently preparing that for a clinical study in the European Union. We're working with our partner contract development and manufacturing organization, which is an outfit called BioVaxys, and they're based in Lyon, France. We're working together now to build out the GMP bioproduction facility and to validate the production capability. We expect that to be completed by the end of this year, and in parallel with that, we should be ready to submit what's called the CTA, that's the European equivalent of the IND in the US, late this year. Our plans for the clinical trial itself are for that beginning early next year in Europe. And we're doing that together with our Spanish pharmaceutical partner, ProCare Health of Barcelona. We're going to be collaborating together on the clinical program development program, and ProCare will be marketing the vaccine in the European Union once it is approved.
And very exciting, because the treatment right now for ovarian cancer is not where we would want to see it. It's really debilitating, and at this point there is not a lot of treatment options. So this is really, to me, groundbreaking, because if we have more treatment options for people that have ovarian cancer, this could really change the course of, I imagine, probably the symptoms they may be experiencing, longevity after being diagnosed. And I haven't heard of anything like this before. Why target ovarian cancer from your perspective? I mean, there's a lot of different cancers out there. Was there a certain reason why ovarian cancer was higher on the list?
Having been a clinician, Dave can probably address the clinical aspects of that question better than I. But in terms of the rationale for us, selecting ovarian cancer as our initial target, you're right. I mean, ovarian cancer is a significant disease worldwide. Over 250,000 women are diagnosed with it annually. It's the leading cause of death from gynecologic malignancies in the U.S. In terms of the numbers of untreated patients and the severity of it, it made a lot of sense for us to target it. The other factor involved here is that the drugs that are currently used, the chemotherapeutics, they're marginally effective, and so there is an opportunity here for new therapeutic modalities.
Our technology, whether it's haptenized viruses or haptenized cancer cells, are platform technology. So although we're choosing certain diseases to start with, they're theoretically applicable to other diseases. So we could target other viruses, and we can target virtually any kind of cancer. Most of the work in the past, as I've mentioned, there's a lot of work that we did when I was in Thomas Jefferson University, and also a company that started from there. There's a lot of clinical work, clinical trials with the prior Haptenized vaccine. Most of the work was in melanoma and in ovarian cancer, and the results were very promising. They were not controlled, randomized controlled trials. They were single-arm trials, but the results were promising and highly suggestive of improvements in survival in both melanoma patients and ovarian cancer that received the vaccine. So we've chosen ovarian cancer because we have some experience suggesting that it's going to work in that cancer. And secondly, because it's a terrible disease, as are most cancers. And thirdly, although chemotherapy can produce very good results in maybe 20%, 25% of patients, the other 75% do not respond and die of the disease. So there's a lot of room for improvement. So that's our reason for choosing that. If we're successful in the future, our intention is to go after other cancers. We have kind of in our mind a list, but certainly we're still interested in melanoma. Colon cancer is an excellent target, kidney cancer, but we could do virtually any cancer. It's just a matter of the resources and having the time to do the work.
And the potential there is just, it's hard to even imagine, because if you're able to use this, as you said, this platform for ovarian cancer, and then can move on to other cancers for treatment, prevention, so many different aspects there that we've touched on, that can really be life-changing and could be a whole really new branch of medicine when we're looking at it that way. In our last couple minutes, I did want to touch base with COVID-19, and we talked about that a little bit at the beginning, but what is the COVID-19 vaccine that you've been developing? How is that different from what we have right now?
The COVID vaccine is, as I described before, the spike protein for the current virus, which is called SARS-CoV-2. We make that protein, or we have it made for us by contract manufacturing. It's got to be made under special conditions so that it's absolutely clean and passes FDA specs, et cetera. But that protein is made for us. We put a haptenone called dinitrophenyl through a process that I described, which is very simple. That haptenized protein is mixed with something called an immunological adjuvant, which is a drug that increases immune responses that many other vaccines also use, and just inject into the patient's muscle. That's essentially the process. And we would probably anticipate 2 injections similar to what you're seeing with the vaccines that are already being used.
And with this type of vaccine, would newer variants of COVID-19 that have been emerging, would that vaccine be protective against those? Like the Delta variant, I know a couple episodes ago, we explored that on the show, the different variants of COVID.
Yeah, well, there are 2 aspects to that. Number one, fortunately, the vaccines that are currently available that use the spike protein use the original spike protein from the original virus that was first isolated in January of 2020, and those vaccines so far have been pretty effective against all of the variants that have come up so far. So we think that our vaccine also would be. However, it is very easy for us if there's a variant that arises that is resistant to the immune effects induced by the old, if I can call it, spike protein. We can pretty easily, without much investigational work, take the variant spike protein and make a new vaccine out of it, and that can be done at a fairly low cost in a fairly short amount of time. So we have just as we have the opportunity to use many different kinds of cancers to target with our Haptenized cancer vaccine, we have the capacity to target virtually any virus protein, even if it happens to be a coronavirus protein from a variant.
Well, thank you so much, both of you, for coming on the show and just sharing all of this insight. There's so much buzz about vaccines right now, and knowing that there are different types that are emerging like this is really encouraging to see, and just everywhere that we're going with medicine. So if people want to learn more, they can go to biovaxys.com. That's BioVaxys.com, and also they're on Twitter for company updates. If you're interested in learning more about the show, you can go to DNApodcast.com. We have all episodes on there, so over 150. And you can interact with us on social media by searching DNA Today. Any questions for myself, Ken, Dr. Berd, you can email in. Email is info@DNApodcast.com. And if you have a moment, we would really appreciate you to rate and review on Apple, because that's how new listeners will find us and can learn more about genetics through the show. So thank you again both for coming on the show. We really appreciate diving into these topics and really getting into the details of them, and really what's on the horizon and what we're looking at being new emerging technology in medicine. So thank you both for coming on the show.
Thank you for having us.
Do you want to influence change in the genomics market? Lucky for you, a new position just opened at Agilent where you can have an impact as a genomics software field application scientist. In this West Coast remote-based role, you will provide technical consultation, training, and education to enable customers to perform data analysis in clinical laboratory environments. You'll partner with sales to serve as the scientific technical specialist to help drive sales and provide support for customers in diverse clinical settings. To learn more about this position and submit your application, use the link in our show notes. You can also access the link at DNApodcast.com. Join me next week on Clubhouse. Dena Goldberg, aka Dena DNA, and I will be talking about how to prepare and what to expect in grad school for genetic counseling. This is on Thursday, August 26th at 4 p.m. Pacific, 7 p.m. Eastern. We'll be answering your questions about grad school live. You can tune in by searching for the Genetic Counseling and the Future of Healthcare Club in the Clubhouse app. Again, this is Thursday, August 26th at 4 p.m. Pacific, 7 p.m. Eastern. Looking forward to connecting with y'all there. You did it! DNA Today has been nominated for the Best 2021 Science and Medicine Podcast Award. If you nominated us, check your inbox to see if you were one of the people randomly selected to vote for the show. I was this year and received the email on August 9th, so pause the show now, check to see if you got the email. If you did, please go ahead and vote for DNA Today. We need all the votes we can get to defend our title as the Best Science and Medicine Podcast. I want to give a shout out to listeners who shared this on social media. Thank you, Kalina, Lauren, Eden, Devin, Amy, Shima, Avery, Natasha, Stephanie, Victoria, Saloni, Lauren, Jessica, Alyssa, Emily, Rachel, Annika, Pat, Jim, Rabbi, Jareen, Leois, Ashlyn, Aneem, Shaeen, Andrew, Jace, and Greenwood Genetics and J-Screen. Thank you so much. Thanks for listening, everybody. Join us next time to learn and discover new advances in the world of genetics.
We're all made of DNA. We're all made of the same chemical DNA.